the cell division to actually take place. So it constitutes about 95% of the total cell cycle. Like the major time goes for preparation, whereas the actual division is the M phase, which only constitutes of the 5%. So you can see how important the preparation is for the process of cell cycle or cell division. So when we talk about this interphase, before this, you people should remember that inside the cell or M phase, right, there are two things that take place. One is karyokinesis and the other is cytokinesis. Karyon means nucleus. So the process wherein the nucleus gets divided into two daughter cells, that is called as karyokinesis. On the other hand, see, first the nucleus divides and then the cell divides. So cell division part is called as cytokinesis and nuclear division part is called as karyokinesis. Clear? So when we talk about the interphase, it has four basic phases. Three major, one is a different thing. G1, S and G2 phase. G1 phase stands for gap 1 phase. S stands for the synthesis phase and G2 stands for gap 2 phase. There is another phase called as G0, which is called as the quiescent phase. So when we talk about the G1 phase, now this is the phase wherein the cell is going to become extremely active in terms of metabolism. But remember, this is the part wherein the cell is preparing itself for the division. But here, DNA replication doesn't take place. Imagine students, for example, this is a cell. Okay. Now this cell has a nucleus inside of which we have the DNA present. Okay. Now, if this cell has to undergo the division process, what is basically division? It's like whatever parts are there in this parent cell, every part will get duplicated and then be distributed to the daughter cell. So the first thing, if you think about it, if the karyokinesis is a thing that has to be occurring first, so in that case, the DNA has to multiply first, become 2 copies, so that it could be distributed into the daughter cells. So that is why DNA replication is something we are talking about. So the G1 phase is the metabolically active stage where the DNA replication does not take place. Here, the next phase is the S phase, which is the synthesis phase. Now here, the particularly DNA replication or duplication process takes place, wherein the concentration of the DNA will increase. And you people know that chromosomes are the physical carriers of the DNA, right? But still no impact will take place on the chromosome number. The chromosome number is going to stay just like that, whereas the DNA number is going to double. So always remember, if initially the concentration of DNA was 2C, that is 2 times, then after the S phase, the concentration of the DNA would become 4C. Okay, but if before this process of the S phase, if the chromosome was diploid, meaning 2 sets were there, so even after the S phase, the chromosome is still going to be diploid. So in the case of animals, DNA replication takes place inside the nucleus, obviously, because that's where the DNA is present. And during this time of the S phase, you people remember centrioles from cell chapter? So those centrioles, they are present in the cytoplasm only in the animals. So they also start to duplicate. Like everything in the cell makes a copy so that it would be distributed equally amongst the daughter cells. Next, we have the G2 phase. So in this phase, the cell organelles will duplicate. The proteins and the different enzymes will be synthesized. Now, let's just say there are few cells in our body that do not divide at all. Or for that time being, the cell is not dividing. So what happens to that? It enters a different phase called as the G0 phase. G naught phase is that all the cells that do not have to divide at that point of time, they will enter this phase, meaning here they will be active in terms of metabolic activity, but they will not divide unless they are needed. Clear?
Yes.
So remember, nerve cells, heart cells, they never divide. So they are always in the quiescent stage. Okay, but few cells, like let's just say the liver cells, they have to divide later on, not right now. They'll stay in the G naught phase whenever they will be needed to divide. Clear? Any doubts?
No.
So now this was the interphase portion. Coming on to the actual division part. First, we will discuss mitosis properly. Okay, I want you all to have undivided attention because it can get a little tricky. So, right now, let's discuss the M phase. So, this is the phase wherein actual cell division takes place, right? And now, whatever has been duplicated, whatever divided during the interphase, now that the distribution process will be done during the M phase. So the M phase is divided into two, that is karyokinesis and cytokinesis. Karyokinesis is where the division of the nucleus would take place. Cytokinesis is where the division of the cytoplasm will take place. Okay. Now the difference in between them you people should know. So cytokinesis, it is a biological process wherein division of the cytoplasm takes place. Cytokinesis happens during mitosis also, during meiosis. And it has no further divisions, it's just one. Whereas karyokinesis is a process wherein the nucleus divides into two. And it is further divided into a lot of stages like prophase, metaphase, anaphase, and telophase. All right. Any doubts?
No.
This is not a boring chapter. So don't be scared. It is very nice. It's very simple and very nice. So when we talk about this picture, see this chart that you see or the pie chart or whatever it is, this as it is can be asked in your exams. They can either ask you to label the part, they can ask you what is the function of that part or what activity is happening in that part. Okay, so you should know that and see how the distribution is there. Like this much is the area, meaning this is the amount of G1 phase time period. Then S phase takes a long period of time because here DNA replication happens. Then G2 is again bigger than G1. And then this part only is the actual dividing phase wherein we have prophase, metaphase, anaphase, and telophase. After this, the cytokinesis happens, which ends up making two daughter cells that are, in the case of mitosis, identical to the parents. So now talking about the actual mitosis. So the other name from which we know mitosis is equational division. So here the parent cell is going to divide into two daughter cells. Both the daughter cells are going to be identical to each other as well as they're going to be identical to the parents. Right? Now, when do we need this mitosis to take place in our body? Whenever there is the need to increase the number of cells in our body. Whenever there is the need to replace the old cell with a new cell. So mitosis is required for all those purposes. Remember that the mitosis is not needed for reproduction in higher organisms. Okay. In those organisms that do sexual reproduction, they don't need mitosis. But those organisms that do asexual reproduction, mitosis can be a method of reproduction. Clear? So the thing about mitosis is that if a diploid cell is undergoing mitosis, diploid daughter cells will form. If triploid cell, triploid daughter cell. So however many number of chromosomes are going to be there, the daughter cells are going to have exact the same one. Now a little bit extra. Mitosis was first observed by Strasburger, name of a person, in the plant cell, and it was also observed in the animal cell by Walter Fleming. And this term mitosis, it was given by Walter Fleming. So imagine back in 1882, the cell division was discovered or named or understood. Now, you people remember the two types of cells, somatic and germ cells? Remember that? No? Yes? Anything? Somatic cells, even if you don't remember, I'll tell you. There are two types of cells majorly in our body. One type of cell, the first one, is responsible for reproduction, and we call them as germ cells. Okay. Second type of cells that are the normal body cells, we call them as somatic cells. So in the normal body cells, mitosis takes place. Clear? So whenever there is the need to increase the number of cells, there is the need to increase the size. We grow in height because of cell division. Understood? Mitosis. Now, mechanism of mitosis. First, we're going to study the karyokinesis part, followed by the cytokinesis part. So, these are the various phases of the karyokinesis. We start with the interphase, which is the preparatory phase. Coming on to the division of the nucleus, we have prophase, metaphase, anaphase, and telophase. If you have problem in remembering it, this is the sequence. The sequence will never change. No matter what you do, you cannot put anyone above or below any. This is how it is supposed to be, because this is how the sequence in which the division actually takes place. So what would you write? I mean, this is a mnemonic that I use to remember. So I played my accordion today. So I stands for interphase, P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. Clear? You can make your own also. You can use mine also. And you know what? From where did I get this? Back very long back when I was even in class 11. I think I was really young. I believe I was in class 7th or 8th. Okay, so I saw this movie. The name of the movie was Sunday School Musical. Okay, so in that movie, the girl, the lead actress, she was a biology student. So she was learning this thing from her father. So she was having difficulty to learn the sequence. So her father taught her the same entire mnemonics. Imagine, I saw this in class 7th or 8th. And when I actually came in class 11, then I realized, oh, I know something that, it's not known by many. So now you people can also use this to remember yours. Okay. But always remember, no matter whatever you use, Hindi, English, whatever, but the sequence is going to stay the same. Clear? You can't change the sequence. Now, carry on means nucleus, and kinesis is movement. Okay, so here it is a process wherein first the nucleus is going to divide. The first stage of the nucleus and karyokinesis is prophase. Pro means first and phase means the stage. Now, during this, the chromatin material, acha, you people remember how in a non-dividing nucleus, the genetic material is entangled, right? Which we call as chromatin. People remember chromatin? We studied in cell chart. As soon as the cell decides to divide, then the condensation of the chromatin takes place and they start and get organized in the form of chromosomes. Right? This is what happens. Acha, one chromosome is made up of what?
Two chromatids.
One chromosome is made up of two chromatids and they are joined together at the center with the help of
Centromere.
centromere.
Yeah.
Centromere. And what is the disc-shaped structure present on the centromere? Remember, the name resembles like the ice cream cone.
Like the thread-like structures that connect the.
This is the chromosome. Right here is the centromere. On the centromere, there is a disc-shaped structure for the attachment of the spindle fibers. What is the name of that disc-shaped structure? Remember when I was teaching?
I know.
Say. The name of this structure resembles the name of the brand of an ice cream. Sort of, not exactly. Acha, I'll tell you the ice cream thingy. Okay, cornetto. Cornetto is the ice cream that it resembles with. Now tell me the name of the structure. I mean, not actually resembles, but still. नहीं याद है? Shall I tell you? Huh? Kinetochore.
Oh yeah.
Kineto. Remember? Kinetochore. Back when I taught you this thing, I said the same lame thing. Now when I'm trying to make you recall, I'm saying the same lame thing again. So remember cornetto, kinetochore. However you want to remember, you use my tricks or your tricks, but you remember the things. So, this basic thing you people should know. Achha. So, first phase, the chromatin material, it starts to condense. Okay. The DNA becomes untangled, and now you can clearly see the chromosomes. You know that every chromosome is made up of two chromatids. Achha, one more thing, students. Like, for example, wait. For example, if this is a chromosome, so this 1 and 2, and this is another chromosome, 3 and 4. Okay. So the 1 and 2 chromatids are sister chromatids, whereas 3 and 4 are non-sister chromatids. Clear? Sister chromatids, they are together. Non-sister chromatids, they are not together. 1 and 2 that I've drawn, sister chromatids. 3 and 4, non-sister chromatids. Clear? So when we talk about this particular prophase, so the chromosomes, they have been formed, but their ends are not visible. Then the chromosome sort of appears like a wall, like this wool ball. Just like how woolen ball is there, same way the chromosome appears during the prophase situation. Clear? Okay. At the same time, the centrioles, they start to move towards the opposite pole. One here, and in the middle, you have the nucleus. So on the either side of the nucleus, the centrioles are present. Clear? Initiation of the spindle, mitotic spindle, meaning now the centrioles will start to synthesize this thing, spindle fibers. Okay? And at the end of the prophase, the cell organelles like Golgi, endoplasmic reticulum, nuclear membrane, nucleolus, these things disappear. Okay? Understood? Yes or no?
Mm-hmm.
So tell me what all things happens at the prophase, one by one. First, what will happen?
Interface preparation will take place. Interface will have
No,
preparation.
prophase. I'm asking about prophase.
Okay. Prophase condensation of genetic material will take place.
Very good.
And then the chromatin will start to define itself into chromosomes. And then
But are the chromosomes clearly visible?
no, they are not
No,
that.
they are not clearly visible. They are looking like a woolen ball. Okay, like tangled up.
And
Then, say.
nuclear membrane disintegrates, and then in metaphase, uh, they align themselves.
No. Metaphase, no. I have taught you prophase only. I'm asking
Oh,
about
yeah,
prophase.
in chapter in school, that's I remember.
So, prophase is the phase wherein the condensation of the chromatin material, this is the most important thing that happens. And how would you see a cell under a microscope and understand that it is at the prophase stage? When you see no cell organelles, right? Because the cell organelles have disappeared. Also, the nuclear membrane is also not there. After that, from the prophase, the nucleus moves towards the metaphase. Metaphase is the second phase. Okay, here what happens when, as soon as the nuclear envelope gets disintegrated or dissolved, then the metaphase begins. So, however many chromosomes were there in the nucleus, now they'll come and line up at the equatorial plane, meaning For example, if this is the cell and these are the chromosomes. So they were randomly present here and there, but as soon as the nuclear envelope disintegrates, now these chromosomes, they're going to, all of them are going to align themselves at one plane, and this is the equator of the cell, the center line, okay? And what they are forming is the equatorial plate, and we call this as metaphasic plate, okay? Which is formed at the metaphase, like the names. Now, when the chromosomes align themselves, and you should remember that amongst all of the stages of the karyokinesis, metaphase is the best stage to study the morphology of the chromosomes. They can ask you this thing. So, the chromosome contain two chromatids attached to each other through the centromere. The chromosome during this stage, during the metaphase, they are very thick and short. So it is very easy to study their morphology because you can make out their ends now. The chromosome condense to form short rod-like structures, right? And then what they do, they start to align themselves. And this process wherein they are aligning is called as congression. Congression means the process of aligning of the chromosome on the equator. Now, after they have aligned, we call it as the metaphasic plate. Now, what happens? The centromere that had the kinetochore present. Now, the spindle fibers coming from the centrioles portion, it will come and attach onto those kinetochores. Okay? This much is clear? So, see this thing. This is the chromosome. So, here. See, here we have the centriole, right? From the centriole, these spindle fibers are arising, the esters are arising, and then the spindle fibers are going and attaching themselves at the kinetochore. Okay, this is what is happening. Now, after that, the anaphase will happen. Ana means up. So now imagine these, like the centromere, which was joining the two chromatids, now the centromere is going to split. So if the centromere will split, will the chromatids stay together?
No.
No. So now imagine, like, for example, this is the kinetochore. I mean, this is the or the centromere, okay? This portion, okay? Attached to it is this spindle fiber, okay? Now, if I say that the spindle fiber is shortening, what will happen? The centromere is going to move away like that. Along with the centromere, the chromatid that was attached to it is also going to go away. So the two chromatids that were together, once this thing, the spindle fibers, they'll start to shorten up. The two chromatids that were together like this, they will start to separate. Acha, have you seen this one thing, students? That whenever, if imagine you people are are somewhere in the air, okay, and you took a rope and you tied it around someone's waist, just like how the horse is tied with that rope, huh? And then you pull the rope towards you. Can you tell me the shape of the person that you can imagine moving towards you?
Starts to rotate.
Rotate, or the person would be like this, like
Okay,
this,
that okay, yeah.
isn't it? If you pull the person will become like this, isn't
Yeah.
it? Can you imagine the thing that I'm saying? They'll go like this, isn't it? So the person will become like this. You're pulling it, the person from here. Same thing happens with the chromatid also. Since the spindle fibers are shortening, the chromatids, they're going to pull away towards the poles of that. Right? So if you see over here, see, this is what I was trying to tell you with the visualization. I was going to take a very long route to give you the example. I was going to go for Spider-Man and the web and everything, but I didn't. So now you see how the shortening of the spindle fibers makes the chromatids look a V-shaped structure, right? So this, like the shape of the chromosome, can be best studied in the anaphase stage of the cell cycle or the mitosis. So let's see what happens. So ana means up, stage, phase means stage. So the centromere, which holds the two chromatids together, they split up. And now they get separated, and these chromatids that are slowly moving away, or the separated chromatids, they are going to be the chromosome of the future daughter cells or the daughter nuclei. So the spindle fibers that were attached to the kinetochore, they will start to shorten, and the daughter chromosomes, they're going to migrate towards the opposite pole. One would go the other way, the other would go the other way, like opposite poles, right? And in between them, these dotted lines that you see, these are representing the interzonal fibers. That shows that they were connected initially. During the migration, the chromosome always moves away from the equatorial plate. During the metaphase, the chromosomes were coming at the equatorial plate, but now they're moving away from it. They're moving towards the pole. Okay, so the centromere of the daughter chromosome moved towards the pole, and the arms of the chromosome trail. So during the anaphase, the distant chromosomes would split up and they will form different types of shapes. So the best stage to study the shape of the chromosome is the anaphase. The best stage to study the morphology of the chromosome is metaphase. Clear? Any doubts?
What's the VLGI written?
That is the shape, the various shape. Like for example, if the chromosome is being split like this, it is a V-shaped chromosome. Understood? Like that. Imagine this thing. Do you people remember metacentric, submetacentric, acrocentric, different types of chromosomes? Remember, depending upon the position of centromere. So for example, if I take this chromosome, which has a very long leg, and this thing. So in this case, when they are going to split up, it's not going to form a V. Rather, it will form an L shape. So depending upon the type of chromosome, when the chromosomes split, the centromeres, the chromatid move towards the opposite poles, they form different shapes. And that shape can be studied best during the anaphase. Clear? Understood?
Yes, ma'am.
Now the last phase. Telo means to end and phase means the stage. So telophase is that stage wherein the nuclear division or the karyokinesis is coming to an end. Now the chromatids, they have reached their respective poles. The mitotic spindle disappears. After reaching the pole, the chromosome becomes uncoiled and they become thin, slender, and lose their identity, how they used to be. Decondensation happens, now they become chromatin again. Nucleolus, endoplasmic reticulum, all the cell organelles, they come back. Nuclear envelope also come back. So now the situation is that if this is a cell, we have nuclear membrane here, genetic material. Another nuclear membrane, genetic material, meaning the chromosome, right? One cell contains two nuclei, but that's not what we want. We don't want a binucleate situation. Instead, we want to form a daughter cell. We want to divide it into 1 and 2. That's what we want. So basically, the process after the telophase is the cytokinesis. Okay, but see over here, the various stages which we have just talked about. So here you can see how early prophase, you can't make out the ends and the beginning or anything, right? Then it undergoes the condensation process, which forms chromosomes that you can still clearly not see the chromatids. But again, you see how they are little bit arranged as compared to the first situation. Third, when they are moving towards the metaphase, slowly they are transitioning towards to come at the equatorial portion. Now, finally, they have formed the metaphasic plate. Then the chromatids are going to split. I mean, the centromere is going to split, and the chromatids are going to move towards the opposite pole, right? So this was one chromatid, this was another, this was another, and thereby the chromatids are reaching the opposite pole. As they reach the opposite pole, now you cannot see their structure or anything. Now they'll go back like this. Right? Something that you cannot make out. But still, one cell has two nuclei. So we have to divide that and form two cells. right? Cell means the plasma membrane plus inside that nucleus and everything, right? So after that comes the cytokines. So cyton refers to cell, right? And kinesis again means the movement. This is the last part of cell division process wherein the cytoplasm will divide, thereby forming the two daughter cells. Now, what would happen if the cytoplasm doesn't divide? This mostly happens in the case of plants. Okay, remember it as a rule that every time karyokinesis, it has to be followed by cytokinesis, always. But what would happen if the karyokinesis is not followed by cytokinesis? In that case, multicellular situation will be there. I mean, not multicellular, multinucleated situation is going to be there. One cell, multiple nuclei, right? So that situation is called as syncytium, single cell containing large number of nuclei. So in these organisms, the division of, like, whenever the karyokinesis doesn't get followed by cytokinesis, so what happens? Thing that happens is called as a formation of syncytium. This usually happens in plants, very less, like, number of times in animals, but mostly in plants. Now, the cytokinesis of plants and animals is very different. The karyokinesis is same, but the cytokinesis is different. Acha, see this. For example, this is a cell. Okay, this right here is a cell. Now, this is one part where the nucleus is, and this is another. Now imagine it pinching off like this, and now you have two cells. This was situation number one. See again. Cell is there. Pinching off takes place to the middle of the portion of the cell. So it pinched off and now you have two cells. Second situation is when it is right in the middle and something would cut from here, like move from here like that. So the difference between the cytokinesis of a plant cell and the cytokinesis of an animal cell is the way the cell splits. In animal cell, cytokinesis is achieved by the formation of a furrow. Furrow means deepening, like this, deepening. Then it will separate. Okay, so the furrow in the plasma membrane of the cell deepens, like from here. The force that acts at the center is called as centripetal, right? Towards the center. Ultimately, it will divide the cell into two daughter cells. Thereby, this is furrow formation in animal cells, which takes place centripetally. Centripetal movement is there. Whereas, if you talk about the plant cell, so here furrow formation will not happen. Instead, cell plate formation will take place. Cell plate will happen, will be formed between the two nuclei that are there, and the cell plate phragmoplast is formed, which is like a cell plate only, and it grows centrifugally, meaning towards like that, outer side, not from the out to in, from the in to out, thereby forming one and two daughter cells. Clear? Because plants, they have an extra layer of cell wall as well, which is not found in animals. So the difference between the cytokinesis in plant cell and the cytokinesis in animal cell, you people should know. Plant cell cytokinesis, it happens by the formation of cell plate. Animal cell cytokinesis, it happens by the formation of furrow. Furrow means deepening. Cell plate formation starts in the center and then grows outwards. This is a centrifugal kind. Furrow formation starts at the peripheral portion, moves towards the inside. Yeah. Any doubts? Then coming on to the uses, the significance, the applications of mitosis. First is growth. Mitosis is essential for growth and development of multicellular organisms. A fertilized egg develops into an embryo, which finally forms an adult by repeated mitotic divisions. Right? Somatic cells of an organism, they are formed by, meaning body cells, they are formed by mitosis. Second, Maintenance of cell size. In our body, every cell has a definitive size that there is. So that maintenance is done by the cell, this thing, mitosis. So if the cell becomes very huge, it will enter mitosis to reduce the size. Then maintenance of chromosome number, genetic stability. And you know that because here there is no reduction, nothing is happening to the number of chromosomes. So it maintains the genetic stability. Repair. The worn out cells, the destroyed cells, the injured cells, they're going to be replaced by mitosis. Reproduction. Again, like I told you, in unicellular organisms or those organisms that have asexual reproduction, they can use mitosis as their reproduction method. And the last one is healing and regeneration. So mitosis, it produces new cells for healing the wounds and for regeneration. Clear? Any doubts, students? So with this, we have completed the mitosis portion. So tell me one thing. Which type of division is mitosis called as? Wow, very good, very good. Which type of division is mitosis? People have to think so much. Easiest question you'll get stuck on, है ना?
Equational division, right?
Yes, equational division. Okay. All right. Why is it the equational division? Quickly. Why?
Because one cell divides to form two daughter cells.
Two daughter cells that are identical to each other and to the parents, right? Okay. Okay. What are the two phases that happens in a cell cycle? Two major phases.
Karyokinesis and
If-
cytokinesis.
No, that is in the M phase. The two phases are interphase and the M phase. Amongst the interphase and the M phase, which one is the preparatory phase?
Interphase.
How much of the volume or time it constitutes?
$24.
Like time, 24 hours is correct, like human cell divides in that. But this thing, the amount or the division of the cell cycle for which the interphase takes place is 95%. Like the 95% of the cell cycle is interphase part.
Yeah, but then 90 minutes for yeast rate.
95% minutes, percent.
Okay.
Like for example, 100% is the cycle, 95% is taken by the interphase and only 5% is the M phase. Okay, human cell divides every 24 hours. Yeast cell divides in 90 minutes. Bacterial cell divides in every 20 minutes. Okay. So interphase has how many subphases?
3.
Three.
But
What are they?
the G0 phase?
No.
G1 phase, S phase, and G2.
Very good. The cells that do not divide go into which phase?
G0 phase.
Also, tell me, amongst the three phases, G1, S, and G2, where does the DNA replication happens? In which phase?
G2.
G1.
Sorry, S, yeah, S. G2 has protein.
Okay. Now, what are the various phases? Acha, what is the meaning of karyokinesis?
Division of nucleus takes place.
Division of nucleus means karyokinesis. And what is cytokinesis?
Division of cytoplasm.
Okay. What are the various phases of the M phase? I mean, this thing, karyokinesis phases.
Prophase, anaphase, metaphase,
Telophase.
telophase. Telophase.
Prophase, metaphase, then anaphase, and then T. Understood? The sequence is very important. That is why I said. Acha, in which of the following various phases of the karyokinesis does the centromere split?
Anaphase.
Anaphase. Okay. And can you tell me what is a furrow?
It's when the it's when the plasma membrane something splits during cytokinesis in
No,
animal.
no, that splitting is not furrow. Furrow means deepening.
Yeah, the deepening.
Deepening. Okay. Acha. In does furrow formation takes place in plant cells?
No,
What formation takes place in plant cells?
since it has a cell wall, so
Cell plate formation takes.
cell plate. Yeah.
Acha, can mitosis be used for reproduction?
No, meiosis is for reproduction, right?
Think
Mitosis is
again.
for extra
Think
cells
again.
like, to generate more cells.
But think again. Can mitosis be used for reproduction?
For reproduction,
Think I
no.
gave you one exception
Yes.
also. How yes?
Yes, right. Ma'am, it's because they produce two new individuals from
Nahi,
sexual.
nahi. So, but for like normally when we talk about reproduction, my question is, can it be used for reproduction? And if yes, then
Because of
where?
cells,
Unicellular organism, for those organisms that reproduce asexually, why would they need to form gametes? So gametes are not formed. Okay. All right. Okay, good. So mitosis we are done with. Now, once you are familiar with mitosis, then only you can understand meiosis. Because now this has further multiple phases. So you have to be very keen observer over here. But before that, you people write down a little bit. Yes, now you people can write down.
Done.
Done.
Done, ma'am.
Done.
Done, ma'am. Done, ma'am.